you're on Zoom and you're also. You can just stay here. All right, welcome back, everyone. Thank you. Hope you had a good lunch. It is my great pleasure to introduce our next speaker, who needs no introduction, especially to this crowd, but Peter Lopez, who many of you know. Yes, there you go. Peter actually served as president of MetroFlow for several years, and I had the honor of being the vice president while he was in office, and I really learned a lot from Peter on running an organization, and also several conversations on how to run my core better and how to be a better core scientist. So I really owe a lot of my career and my progress in my career to my friendship with Peter. So thank you for that. So he'll be giving us a Peter Lopez retrospective. So enjoy.
Does this have a laser? Yeah.
Yep.
Thanks so much, Tina, for that great introduction. I'm so happy to be here and to see the group, and what I understand, a really high turnout for this meeting, which is fantastic. The group keeps on growing. So I was really honored to have this opportunity. The organizing committee asked Mike, Tina, and the others asked me to come in and give just a retrospective on my career. I think it kind of is a nice fit in with the earlier session where we heard from a number of people who have made headway in different aspects of their careers. So the first thing they asked was for me to generate an abstract and a title, and I came up with a couple of titles, and I was like, that's no good, that's no good. And then I came up with this title, and I didn't come up with this myself. That's why I put it in quotes. So I'll just start out by showing you where this came from.
Walk off. That's what I do. That's what I do. Whoa. All net. All net.
So it was inspirational. I can't identify with one of our greatest presidents here, but I think it's just his inspiration. So let me talk a little bit about my career. You saw the dates up there. I started in 1977. I'm going to go into excruciating detail, and I hope that maybe some of these things might be inspirational. Some things may be informative. I'll talk a little bit of some of the scientific stuff, but it won't be too much. It's really just me taking a walk down memory lane, really self-indulgent, but thanks a lot for giving me the opportunity. So my career's been primarily in core laboratories, and I didn't know anything about core laboratories. Actually, they weren't called core laboratories. Nobody knew what we were actually doing back in the 70s. They hadn't even been named yet. But quickly I realized this is a place where you can see a lot of science going on. You can get involved. You can be kind of, try to be a jack of all trades, which I think was one of my thoughts in all this. Is there a regular mic, or should I just walk this? Because I think it's for the WebEx people. I think everybody up here who was talking earlier knows that these things work best. I mean, everything really kind of in life works best if you can really put yourself out, do the extra effort to help out other people. I mean, it's kind of like the golden rule, right? And in the core laboratory, this is kind of what they call customer focus. It's a fast-paced environment, and it can also be very slow, right? It has its ups and downs. I kind of like that. I found in my career that the core laboratory allows you to be innovative and to come up with, if that's your thing. I mean, that you can be in a core and not necessarily redesign everything. There's job security if you're doing flow cytometry, right? There's not, there's a lot of need, and there's not a lot of trained individuals. One of the things that I think if you're in a core laboratory, it doesn't show up on your resume is that you learn how to be very diplomatic. You have to be able to tell an investigator that their experiment is a PIS, if you know what that means, a And you got to be able to do that and not get them defensive. And the whole point there is not for you to say, hey, you don't know what you're doing, but let's make this better. So you got to work that diplomacy. And sometimes you even get recognition for this, but I think we all know that this is a challenge in the core laboratory because it's kind of assumed we're going to pay you some money to do this stuff. You're going to do it. And that's the arrangement. One of the things that is different, and I put that at the end here, is that I did enjoy my numerous experiences working with engineering teams. And my next slide will hopefully explain that a little. This is something I just got recently. I'm happy and proud to have had support because this is a membership award from ISAC for, I like the part where it says, for those who have achieved peer recognition for advancing the field of flow cytometry. I think that's, and I'm really proud of this, and thank you if you voted for me. Just showing you some, I'm actually going to the end of the slides really fast, and then the excruciating part is getting there, so you'll have to bear with me. Some of you may have gone out and got yourself a Peter Lopez Lego. Thanks for doing that. I have no financial interest with Slingshot. I'm really happy that they selected me to be their second flow cytometry Lego. Johnny Moore was first, and Fred Prepper has come in now, and he's the third. They've got great products. Again, no financial interest, but if you want one of these, get one. If you get two, give one to me because I only have one. And this is it right here. And of course, you can't read that, right? So each one of these is a little time point, a little snippet in my career. And as I said, excruciating detail in all of these. So some of you who are older might know this reference. And if you don't, you might enjoy checking this out. This is something from 1980. And this is it's a cool video. I just thought, and this is the quote from that song. You may ask yourself, well, how did I get here? So that's what I'm going to try to answer. So how did I get here? I'm going to go back to early teenage life and even before that. I always had an interest in how things work, both mechanical and biological. At that point, it's probably more mechanical. And somebody else once heard say this: if you can take something apart, you better be able to put it back together. Okay. And I was always on the taking it apart side, and maybe not quite getting it back together. But eventually, I learned how to do this. I put this thing up here called iFixit. I just became aware of this because I've been doing some consulting for an engineering firm, and they had a kit of tools made by this company. I said, this is a cool kit. I really want to, so I bought it, but the kit came with all of this stuff where this company is advocating, learn how to fix stuff. And if you know how to fix stuff, teach somebody else how to fix stuff. This is kind of something that I think maybe I took up maybe without unconsciously, and maybe flow cytometry was the thing that I was trying to teach people how to fix or how to make work better. I found that, and those of you who know me might think that I'm a bit of a gearhead, and that came from some things that I had done in the automotive side. That's a Chevy Impala Super Sport 1963. I worked on, it's actually the right color. My older neighbor across the street had this, and we used to get into this 327 Chevy engine, trying to get more horsepower out of it. You can probably imagine, maybe you don't imagine why you'd want more horsepower. That's so you can go faster, and then you could go and race your friends. And this is continued, and this is my current project, which is a 1983 Pininfarina, which is actually a Fiat. And those of you who know the acronym, Fiat means fix it again, Tony. And so guess who's I'm Tony in this, so I'm fixing it again. But it still runs. That's the car parked at Metro North where I catch the train. So I was going into work. I also worked in a restaurant. This is Francesco's Italian restaurant. I worked here from 1971 to 1976. And I started as a dishwasher and worked my way up to becoming the weekday night chef. So I think this experience was kind of helpful for me because this can become a very frantic environment. we used to say the bus just let out. That means a bus just pulled up because all of a sudden 50 people walked in. And you got to be able to make that happen. And in a way, core laboratories can sometimes have that kind of dynamic. I was an astronomy nerd. Maybe a lot of you probably don't know that because I don't talk about it that much anymore. But later in life, I started traveling to see solar eclipses. I will, how do they say that? I will travel to eclipse, or I will drive for eclipse, whatever. I'm going to go see the next one, and I'm not going to see it where I'm actually going to Mexico to see it, hopefully, because the conditions, they have less of a cloud cover during that time of the year. But I built a telescope. This is not my telescope, but this is almost exactly the same thing. I built that at age 16, so I learned optics. I built, it wasn't a kit. I designed it and built it myself. And then it's still around, by the way. It's sitting in my shed, and at some point I should take it back out again. And I was interested in photography. So these things, were interests of mine. And then, all of this, I was interested in computing too. This is one of the computers that people are not, this thing is this big. It's tiny and it's flat. That was released in 1980. It didn't really do a whole hell of a lot. This is when Apple was making the first Apple computers, and I don't even know if there was anything on the PC side. The PCs didn't exist, right? But I bought this thing. I thought it was cool. You could take it apart. See, take it apart. You could take it apart and put it back together. And if you were interested in that, and I learned a little bit of computing at that point. So these are all things that were on my plate already. I was just playing around, dabbling with this. So I later went to school and in Syracuse at Upstate Medical Center, and I studied cytopathology. So now into the biological point here. And one of the things that I'd like to point out to this group. Where I found this to be a very big advantage is I really learned microscopy, because cytopathology is basically just peering down a microscope and diagnosing cellular material. And I really recommend if folks here are not using microscopy, if you don't know where there's a microscope, find a microscope and get comfortable with it, because this is going to help you with your flow cytometry. Because what you see on the flow cytometer, okay, there's imaging flow cytometry, I understand that, but not everybody has that, right? So if something's going wrong or something's odd, take that sample and put it under the microscope. You got to know how to use the microscope. You got to be comfortable with it. So I basically learned how to look at images like this and do diagnostic work. So that's my history. That's leading up to the first point. So as I said, excruciating detail, folks. This is the crap. Going by itself. See, it wants to get done quick too. So my first step was a fortunate recruitment, because I didn't have a job after I trained in cytopathology, frankly, because I was kind of okay at it, but it seemed to be not, I wasn't destined for this to be a long-term career. I think I knew that. I think my professors knew that. So I didn't have a job. And Leon Wheelis, and I'm going to talk a little bit about him also, came over to me while I was peering under the microscope, and he tapped me on the shoulder. He said, "Would you like to come work for us?" And I knew what Leon was doing, because I did, after my internship, part of the program was to actually intern and be in a laboratory doing traditional cytopathology. Rochester had this other project going on where they were basically trying to, as they say now, automate me out of a job. They were trying to do what I had learned automatically using a flow cytometer. Now, I wasn't thinking that far in the future. All I knew was I'm learning this biology stuff, and they've got optics, they've got mechanical stuff, they've got lasers, they got computers. I got to go there. So, but I didn't have any reason to go there other than the fact that Leon Wheelis, who was my first, well, maybe Stan Patton also, but one of my long list of mentors, gave me a break because I didn't have a job. He said, "Why don't you come over and work with us?" And I said, "Hell yeah, when do I start?" So what we were doing was we were trying to automate slit scan. We came up with a technology. I'm just going to go into this a little bit. So this is going to be a little bit on the more less me and more scientific presentation. But let's just talk about Leon. Leon Wheelis. A lot of folks here probably are not aware of this person. I'm going to bring up a lot about him because he really helped me out initially. Leon was one of the presidents of what we now call ISAC, okay? And he presided over, it wasn't always ISAC, it used to be SAC. Initially, they forgot about the international component and they just called it Society of Analytical and it was a Society of Analytical Cytology was the original name of this group. Later, it was rebranded as we do with everything nowadays. And in 1982, Leon ran this meeting. But of interest is Leon was one of these early pioneers. And I'll tell you, it's really important to know what these folks were doing and what they accomplished, because we do a lot of stuff now which we've learned from somebody else who's learned from somebody else who's learned from somebody else who's learned, but it started here. And everybody knows about the telephone game, right? Things get translated, mistranslated and miscommunicated. It's really good to try to get back to this stuff and see what this, because all right, so you got the communication breakdown. Okay, Led Zeppelin, right? But also, it's good to know how this stuff evolved and where we were back then, because you got to know history, or else you're gonna, you can make mistakes. And there's Leon. So that's my first boss. But there are other luminaries here. I mean, Myron Malaman was the first president of before it was even SAC. He was number one. That's Scott Cram, part of Los Alamos, one of the first people to sort chromosomes in bulk before the Human Genome Project ever happened. Scott was part of a team sorting chromosomes. And you wouldn't be sorting without this guy. This is Mac Fulwiler. This is the guy who invented cell sorting. Okay, so remember, it was Mac he invented cell sorting by thinking outside of the box. He took two things that were two separate items that you would never, other than Mac, would think of putting in together. He took inkjet printing and Coulter volume and said, wait a minute, inkjet, well, maybe it doesn't have to be ink. Maybe it could be saline. And maybe in the saline, or PBS, we have cells. And Coulter volume is what we have for making measurements. There weren't people doing a lot of fluorescence measurements. Everybody always refers to Lee and Len Herzenberg for inventing the cell sorter. They invented the fax. They invented the fluorescence activated cell sorter. They didn't invent cell sorting. He did. Brian Mayle is the first editor of the cytometry journal, the first journal of flow cytometry. And then there's others. Tom Joven described energy transfer. If anybody's doing energy transfer, you might have him on your reference list. So that's where that came from, and others. So I'm going to go a little bit through a slit scan. This is where I got my start. I think there's some stuff here that you might find interesting. It's a method of obtaining higher resolution measurements from cells in flow. Today, the answer to doing higher resolution in flow is to get an image. Okay, we have imaging systems. We were doing it without imaging, not because we didn't know how to do imaging. We made the first imaging flow cytometer in the late 70s. So I'm going to show you some pictures. People know about Amnis and imaging, and that we had a machine that we used, but we built it for a specific purpose, which I'll tell you about. But the point here is that all flow cytometers, unless I got that wrong, and please correct me, but all flow cytometers focus their laser beams to a point, and the cell is passing through that point, but that focus is bigger than the cell. So you have a cell 10, 15 microns in diameter is going to go through a focus of 30 microns. So you're just getting a pulse of light. The machine doesn't know what it's looking at. It just sees a pulse of light. Difference here in slit scan is the laser was skinny down and made into a plane of light that the cell would be passing through, and it was much thinner than the cell. It was a very, very clever technique, okay? So what was neat about this, and again, I can talk about, this is my history because I was involved in this. We first, and it's kind of just something to keep in mind, you got to walk before you can run. We first demonstrated a proof of principle that slit scan, that slit scan actually can do something informative. We did it without going into flow. We did it on a microscope. So I remember I note microscopy is an important adjunct or something to be comfortable with. So we made a microscope that would take slit scan data. I'll explain it a little bit more in the next slide. But after we found out, and my job was to say, this is a cell, I've looked at it, it is this type of a cell, and then we did a slit scan and that entered in the database. No AI, no machine learning. This was seat of the pants kind of learning. And we built up this big database. And then we said, if we analyze the data coming from the slit scan, the pulses that were being generated, can we say anything diagnostic in this? And the answer was yes. And we said, okay, if we can do it on a microscope, let's move it into a flow system, built a slit scan flow cytometer. And my job was to pre-diagnose samples. And these were flow samples. And these were pap smears, basically, which usually are on a glass slide. I would look at it, we'd put it on the machine, and see and let the machine classify every one of the cells and decide, is that cell cancerous or not? So that's the goal here, is to say, is this or pre-cancerous? And what we found was that the system had an unacceptable false positive rate. Well, crap, I keep on pressing. Take this away from me. I'm hitting the wrong button. What that means is that we would put samples on that we knew had no cancerous material on them, and it would say, occasionally it would say it saw some cells. So we're like, what's going on? And the answer the engineers came up with, Peter, if you looked at it and you don't see anything on there that looks cancerous and you flow it and it sees something, it has to do with the flow. And how are we going to find out anything about that? Let's image it. So we built an imaging flow cytometer. So that was it's a stepwise process, but again, illustrating how you can build on earlier knowledge. And the imaging system showed us that cell orientation of these flat squamous epithelial cells, they're the skin cells, basically, they're flat. They can flow in different directions as they're going through a flow cytometer. The orientation was what was key. So the engineers came up with a solution. And again, you might look at this and say, is he nuts? We built a 3-dimensional flow cytometer that I was operating in 1980. So this was looking at cells, imaging. I mean, it wasn't imaging though. We found out from the image what we needed to do. And then what we did was we built a 3-dimensional flow cytometer. And what it was doing was it was looking at these cells and figuring out in 3 dimensions, which one looks best. And you can imagine if you got 3 dimensions, one of these detectors is going to see the cell in the right way. This is the data, and these are images coming from the slit scan flow cytometer. These are pulses of light. You see, and these lines that are drawn. This is real-time classification that was done from that pulse and determining how big the nucleus is and how big the cytoplasm is. There's no antibodies here. This is done with acridine orange. It's just a DNA, it's a nucleic acid stain. And what we can see here is that when cells would go through two side by side, it would, this kind of a pulse is a bad pulse. It thinks that's a tumor cell, but it isn't. The only way we could find that out was to image. And this is just showing you some of the, so this kind of gives you a little bit better impression of how slit scan works. The cell goes through a plane of light and you get a pulse. On today's flow cytometers, that's just a pulse. I mean, we all know about double discrimination and things like that. It's just a pulse. You don't get any shape, okay? And the easiest way to get shape is to just skinny down the laser. Okay, so let's move on. So now we're moving on to this one. I know nobody can see this. This is Fox Chase Cancer Center, Philadelphia, 1982 to 1986. So I moved to Philadelphia from Rochester. That other work was Rochester, New York. I got to work with a wonderful immunologist, Don Mosier here, seen in the 1982 Fox Chase, what do they call it? Volume 3. I guess we had some numerous volumes of this. Don is a very famous immunologist. He helped us understand antigen presentation, among other things, B cells. I mean, if anybody's doing B cell work, you're gonna probably see references with Don Mosier. He's a wonderful mentor, and just included some pictures here of what lasers look like. That's a laser. We don't have anything in there as a size indication. I put this picture in here because that's kind of cool. That's the front end of it. This is all glass. This is glass. Actually, the glass continues. This is a magnet structure that goes over the glass. The glass, it's a beautiful, beautiful, delicate item. And the glass goes from here to here, and it's vacuum. There's a vacuum in there. It's a giant vacuum tube. This is what lasers looked like back then. Okay. What do I have up there? Okay, so it was a 2-laser fax 2 is what I got started on. I later modified that by hand. I actually went in because I thought I could do it, and I did it. I added another channel to it because all of a sudden there was another color that was available back then. This really quick story, Don Mosier, it was a well-respected immunologist, pretty much cutting edge on a lot of stuff. And he got a sample of phycoerythrin. And back then we didn't have CDs. I think this was on like Thigh 1.2 because he was a mouse immunologist. So he's doing it. So it's a T cell marker. And he said, Peter, let's go ahead and try this out. And I had the spectra, absorbance and emission. And I looked at it, and we didn't really get much more instruction than that. And I just ran it, and we had a 2-laser fax 2. I'm going to show you. I'm going to skip ahead because I'm probably going too late already, too slowly already. But because it's probably here. No, it's not. Oh yeah, it is. I'll get back to that story and remind me because I'm getting old and I forget easily. But anyway, I wanted to point out, and this actually gets to the point. This stuff that's highlighted here, it's hard to see, so I blew it up. So in 82, Fox says the most advanced model cell sorter can identify and analyze. Now look at these numbers. In 1 minute, 300,000 cells. Do the math, right? 5,000 a second. Wow. This was big time. The area's first cell sorter with dual laser beams. So on the East Coast, this hadn't been seen. Don was a high enough powered immunologist that he had this. So he had a 2-laser. And why do you need 2-laser? So it can perform 2-color sorting. Wow. Just so you know what that's all about, there were only 2 fluorescent dyes that were readily utilized at that time, fluorescein and rhodamine. And rhodamine and fluorescein need different lasers. So we had one of those. This was hot stuff, right? And Don was way advanced because he had that. When I walked in, that facility was 2 years old. He started this thing in 1980. So my thought when this phycoerythrin, and I remember what I was talking about. My thought with the phycoerythrin was, oh, they want us to run this because we're one of the few labs that have 2 lasers, and so one laser's got argon gas, the other one's got krypton, and krypton has a line that's about 561 or 568. Do you remember? 8. Thank you. 568. He knows this stuff. The 568 laser is just perfect for phycoerythrin. So I said, cool. And you had to manipulate the lasers to make them do this. I set it up for 568 and I ran it. And this stuff is screaming bright. I said, Don, this stuff's fantastic. Let BD or Herzenberg know. And then he went back and he told them that, and they came back and said, you didn't do it right. I said, why didn't I do it right? They said, we want you to excite it at 488 and see what you get. It's like, why would you want to excite phycoerythrin at 488? Back scan, right? Now all of a sudden that you got one laser can do, the other colors. So that was kind of the preface to that. That's the machine. That's the monster. That's the fax 2. Just to let you know, these things over here were always very different from lab to lab, and there weren't a lot of these around. But that was the first commercial flow cytometer that I used. This is the back of one of these things. I just wanted to point out, there's the laser, big thing, right? And these are photomultiplier tubes. And yes, there are only 2 there. This is a 1-laser fax 2 and 2 PMTs coming off of the 90-degree side. What are you going to get? 1 color and side scatter. So that's a 1-color machine. Interesting point. Back in those days, a lot of the data that you see in the literature from flow cytometry data was actually done on cell sorters because analyzers weren't that prevalent. I mean, the big deal at a university was to get yourself a cell sorter. So I'll talk more about, I don't have a timer. I just don't want to go way over. Anyway, well, let me point out something about that system. And this may be a surprise. I'm guessing this is going to be a surprise. I was doing not what I was doing, but this story. I was doing in the 80s. I was doing chromosome sorting because Don was great, and there was a really long leash. He let me do whatever I thought was cool, just as long as I could tie it into some mouse immunology. So I was sorting out chromosomes. I designed a prep to sort out chromosomes. I actually modified that machine, that VAX2, optically to have a tighter beam focus. Remember, I did photography and astronomy and stuff, so I knew how to do that. And what I found was this is based on DNA label. This was ethidium bromide at the time, which is kind of like propydiamidite, although Kelly will probably scream at me for saying that. But anyway, you would get multiple peaks, and these peaks, in this case, represent groups of chromosomes, but you could sort them, and then you could study individual chromosomes, okay? What I found was that on the FAX2, if I had the forward light scatter detector filter in place, I got very poor resolution on the low end, the dimmest, the smallest chromosomes. If I took the filter out, they went away. So I went to BD and I showed them this data, and they said, what's going on? And I said, well, I think I can see it. And what you could see when you put that filter in, optical filters like neutral density filters kind of act like mirrors. And when you put that forward scatter filter in, the laser would hit that, go to the forward scatter detector, but some of it would bounce back and it would go right back onto the stream. And that was what was causing this. So with BD's help, BD came in and they said, let's do something different. Let's tilt the forward scatter detector so that the light hitting that filter will bounce back, but not hit the stream. And it worked. And guess what? Every BD instrument to this day has a forward scatter tilted detector based on this data. Now, I'm not making, this is just like, I want to bring this up because I've retired and I was cleaning out my office and finding a lot of stuff that was all in packaging that I hadn't touched on. I just took these pictures recently, and they're in color. You can see the kind of the yellowing going on here because I had the data. And I was like, oh, I forgot about that. But it's kind of a cool story. But again, it just shows the types of things that you can do in a core laboratory environment. So let's move on. Oh, 13 minutes, crap. All right. Yeah, I'm going to blow through this as fast as I can. Dana Farber, Harvard. I worked with Ellis Reinhardt, this guy who kind of worked out the T cell receptor function. I helped with that with using Coulter equipment. Again, we were doing, we're trying to understand T cell function. We were doing a lot of calcium mobilization assays, but that's analytical stuff. And we were still using cell sorters at the time because the cell sorter had the lasers. In this case, it was the UV laser that was needed. I was trained as a BD biomedical engineer, which is a short-lived program. BD put it out there for a while. I thought it was really cool, and I was able to go where they taught you how to fix your own instruments, and then they said, "Whoa, we're losing service contracts." So they made the so makes sense, right? I did crazy stuff with Ethernet. I ran my own subnet, which was Harvard didn't even know I was doing this. They just gave me some IP addresses, and I ran it. And I was backing up data for all my machines and putting it onto a microvax. We didn't have PCs. We didn't have whatever you did with computers back then was with a computer suite. Somebody they had all this equipment. And at that point, I also moved into some deconvolution microscopy and brought in an instrument. So I started running multiple cores at that time. And came up with a couple of, I think, very important publications. One of the first hematopoietic stem cell isolation protocols. I'll just show you. That's this. Guess which one I am. So that, yeah, I needed a haircut at that point. But there's some folks from BD who are here. Angie actually was very influential in the design of the Vantage, the BD Vantage system. I think that's right. Steve Schleifer, Dave Small. These are the BD folks. These are all we were here for fact scan training. Interesting thing there also is back then, an instrument like the fact scan, at least in my lab, we would run it. We wouldn't let people walk up to analyzers. That was a very big change at that point. So this is about 86, 87 that people were saying, hey, let us run it. And we would say, no, you're going to break the machine. No, you're going to break. This is a Boston Globe paper. There I am. Oh crap! I did it again. Sorry, folks. Oh, right, we're going way ahead. Well, that's what we want to do. We want to move through this. This is one of the first stem cell isolation protocols that I was happy to be involved with Dr. David Scadden, who was actually not at he was at the time it was Deaconess Hospital. Now he's at Mass General, and we worked out a protocol for doing a stem cell hematopoietic stem cell isolation, and also I got to work on one of the first apoptosis detection methods that the thing that was used at that point was called a TDT assay, terminal deoxynucleotidyltransferase. Has anybody used that? If anybody uses it, they're going to do this because that protocol sucks. It's just really difficult. Multiple fixations. You start out with 5 million cells. You're lucky if you get 5,000 after it. So this is one of the first. One of the things that I want to point out in that paper, which was basically using Herxt, we identified what I think everybody now takes for granted when they want to do viability staining using DAPI or something, that there's 3 populations in there. And we identified the fact that this population is the apoptotic population. So we used to use this as an apoptotic indicator. We had a protocol. This was lost to history, but it's from 1992 that we came up with this protocol. Okay, skipping ahead. So now I'm thinking, well, maybe I got to try out industry for a little while. So I went over to Cytomation. Again, these were all recruitments, by the way. The people just would say, why don't you move to Fort Collins, which I did. I moved to Colorado for a number of years. I was employee 30 something. Somebody knows that number, but I don't. But this was the first the company was called Cytomation. This whole product line was picked up by Beckman Coulter. And we had the first commercially available cell sorter designed from the ground up. There were competitive systems out there. I'm not trying to dismiss that, but this system was built from the ground up to be a high-speed cell sorter. And there's some history there. That could be a discussion over some drinks if you want to hear about that. It ties in with the Human Genome Project. It's kind of convoluted, but it's true. And I did some stuff for optimizing that system, including a sheath fluid filtration system, because the thing couldn't maintain drop delay when I started, and I fixed that. And then I came up with a system that we called the Z configuration. I wasn't supposed to do this. I'm not an optical engineer, but I knew enough about it, and I knew enough about their design that I thought that this thing wasn't sensitive enough. So I redesigned the PMT layout, and then that turned into a product. And I gave talks and stuff. And also at that point, I was a co-author of one of the first biosafety guidelines for cell sorting, mainly because nobody else was doing high speed. So they brought me on board for the high speed aspect. Big lasers, MetroFlow, all kind of history. Got recruited back to New York specifically by David Ho. He called my house, and my wife can tell you the story because I didn't realize who I was talking to when he called me to try to convince me to move to New York. But David Ho is Time Man of the Year. This is the individual who turned HIV/AIDS into from a death sentence into something that could be managed. And I'm happy to still. He just recently retired, and I think his retirement is going just as well as mine is, which I'll tell you a little bit more later. Then got recruited by a bunch of my old friends from Good to Keep. Don't burn those bridges. Well, from your previous, I had worked with Mike Dustin, a world famous immunologist, Steve Burakoff also. At Dana-Farber. They had moved to New York. They had moved to NYU. Dan Lippman, another well-known, and people know that name, right? So these folks started recruiting me. It took them 2 years to convince me to move from Aaron Diamond to just go across the street, because Aaron Diamond is across the street from NYU. And in that location, I had to set up a new laboratory, BSL-3, which was promised to an investigator, and that investigator was ready to bail unless we got this. So I had to do that fast. I did that in 2010, 2011, set up the whole thing. And then they moved it, which is also interesting. I've got a lot of history moving BSL-3 laboratories around. As Tina pointed out, I had some executive roles, including MetroFlow, and these are the ABRF is kind of another organization that I think people might be interested in. These are regional organizations. I collaborated with the Monterey Bay Aquarium Research Institute on specifications for putting flow cytometers into things like this. This is an autonomous underwater vehicle. This is basically a robot that can go out and do missions. And we were trying to spec out how to put stuff in that payload area that would do minimal flow cytometry, but do it in real time, send telemetry back. You don't have a lot of battery, this is out by itself. You got to think about battery power and stuff like that. I was lucky to have that opportunity. Did some work with centrifugal elucidation. I think people should have some familiarity with this. This is a non-label method for purification of cells based on cell size. And you can in many cell types, you can pull out cells in different phases of the cell cycle. With this, you can enrich. And if anybody's studying anything related to cell cycle phase, they usually have some other method for synchronization. And the question that everybody should also always ask is, how does your synchronization process affect your results? And everybody's going to say, well, they're going to hem and haw. If you use centrifugal elucidation, it's just a centrifuge. We relocated our lab. Joined the board of directors for the New York Blood Center. That again was another recruitment. But the last point here I want to talk about, and I highlighted that because this is, I'm going to kind of say this was, I'm not going to say my crowning achievement, but I think this is one of my largest contributions in my mind to the field. And this is something that we identified, and I keep on messing this up. From other people's studies, people were pointing out to the fact that cells coming off of a cell sorter didn't perform well in metabolic with their metabolites. So we reproduced this work in our lab in direct collaboration with our metabolomics core. In fact, they drove most of this work. And this is an interesting thing with core laboratories where if in environments where all the directors are talking to each other, you can do some cool things. If you hear from one lab, I've got a problem, and they're communicating with you, you might be able to come up with something, or at least study it with a little bit more detail. And what we did was we basically cell sorted, and we had controls, and we did a very large metabolite screen. There's lots of them on here. If you're into metabolites, yours is on there someplace. And in conditions where cells were not sorted, these numbers are increasing one-fold, two-fold, and decreasing. We were seeing things either staying steady or maybe increasing a little bit if you didn't sort. But once you sorted, everything dropped. All of these metabolites just went down. Okay. We know we can sort every day. We sort every day. We're not killing cells. But the point that I think we're trying to make is you're not killing them, but you're removing all of their metabolites at some level. We figured out how to fix it. And we figured out how to fix it because we're lucky at NYU to have this system, which is a dual-headed system, a two-headed cell sorter that Sony made. Initially, this was made by Gary Durek's company at iSight. This was initially called a reflection, and then Sony bought it and they modified it. It's a really cool tool because you can do two different experimental conditions simultaneously. And in metabolomics, time is up. You have to have, if you want to compare things, they have to have the timing being exactly the same. So we were sorting simultaneously, and we sorted on a hunch that I came up with from knowledge from other experiments that we did with elucidation, where we found that the buffer makes a big difference in your results if you're doing metabolite work. And the way we fixed it with the elucidator was we ran the cells in with a buffer that is exactly the same thing that the cells were in, basically complete media. Everything, all the goodies, complete media is what we would run. So I said, let's try it in cell sorting. Nobody wants to do this. This is a PIA, right? You have to make sure you're running complete media through the system. Luckily, we could do that, and on this system, we ran one. It's the same sample split into two. These are two sort heads in this unit, with PBS running on one, RPMI running in the other. Oh crap! And we use different collection medias because collection media has been thought to improve things. I didn't show the principal component analysis here. Bottom line is, if you have a cell line that's giving you a whole lot of trouble, you might want to try what we call complete media as sheet. We call it CMAS. It's a pain, but it could save an experiment. So just really briefly, the end of that part is Usually people worry about cell sorting. Did it kill the cells? Yeah, you don't want to kill the cells. Okay. And this is just one, there's a lot to cell sorting where you can do damage to cells, but this part we believe is common to any droplet cell sorter. Droplet cell sorters seem to do this because we did this on multiple machines. We did it on the MetroFlow and we did it on that Sony machine, and we got the same results. The thought here is if your cells are super happy, you got 100% viable cells and you put them on a machine, if you believe our work that 6 is going to happen, that the metabolites are going to be depleted, it may not kill the cells, but they'll be unhappy. But who has samples that are 100% perfect samples up front? There's all sorts of viabilities. So if you layer that on top of this metabolite depletion, what's going to happen? Are they going to die, or are they going to have odd functional changes because they're trying to bring back their metabolites? So we know that certain cells will do certain things oddly after cell sorting, but why? Maybe it's just because they're responding to the fact that they lost most of their metabolites. We looked at many cell types, and others have. So the question is, how does any particular cell type, respond to having metabolites depleted? Is it going to have trouble with antigen presentation? Is it going to have a delay in replication? All these things might just be pointing to this fact. Okay, and if until folks in this room figure this out, because I've retired, so I don't have a lab anymore. We'll talk about that in a minute. To get around this, consider running complete media as sheet. And if you want to know how we did it, it's not in the paper. I can tell you about it. You can do it, and we've done it, and we've actually made some. We've actually had some publications come out with that work. Got that. So that's all. This is supposed to tell you why I got that. And then just it's going to be my final 2 slides. Oh, I'm only over by 35 seconds. We had a lot of examples of different ways that you can make a career, and they're all really good, all really valid. In my experience, an academic flow cytometry course is where you learn your chops, where you build your toolkit, where you learn how to do these things. But it's a difficult environment. I don't know, does anybody here work in a core and think it's easy peasy, right? Okay, I'm not seeing any hands. No, and believe me, folks at higher levels, the administrators, they don't understand this fact. They think that all cores are created equal. Whatever happens in a metabolomics core or a genomics core, probably the same thing. That whole dynamic is the same. Hell no, right? And we gotta strengthen our resolve to get these folks on board, knowing that the flow cytometry lab is one of the most difficult environments in a core. And if you figured out a way not to make it like that, well, share it amongst yourself, because I'm not doing this anymore. And if you are doing it, you're wearing the big S. Okay, if you can make this work, you're Superman, Superwoman. Things that I've done, in my career, you want to make a move, and I've heard people say, "Oh, there's a great opportunity there, but I don't want to make the move." Well, what do you want? Make that. Think about that balance. Are you excited? So I think you get the gist. Can a career in flow cytometry prepare you for something a little different? Well, I think it can, because I've got a different role now. So I retired from my role as the director at NYU's flow cytometry core in February 28th. I was on part-time for a little while. During that time, I got recruited, and this is my new job. And I start this job November 6th. I'm the vice president of technical operations and innovation, and the New York head of the comprehensive cell solutions. It's a long title. I don't have my card yet. And it's supposed to say therapeutic. Oh, that part didn't come. Oh, that part didn't come off. It doesn't matter. That's it. No time for talk.
We'll do a couple questions. Thank you.
I
so
went a
much.
little over, so I'm happy to talk outside. I'm not going anywhere.
Yeah.
Wow.
Anyone have one pressing question they really want to ask Peter?
That's all right.
Okay.
No,
Yes,
okay.
we'll
Yeah,
move
get back.
forward, but he'll be around, and there will be an after gathering. It's informal. MetroFlow does not pay for your alcohol, but if you'd like to come out